So we have looked at different types of chromosomal and gene mutations. So what we're going to do today is look at how these mutations actually manifest themselves in our DNA. And one thing to understand is that mutations happen randomly. So it's not like you just tell your DNA to mutate and it does it. It happens through a series of kind of middlemen, and our message has to be passed on through a few steps. And when this happens, there's more chances for a mistake to occur. So, and what we're going to look at is how the different DNA, if we change a letter, it leads to a different protein, which leads to a different U. So, first, let's look at all the different types of proteins we have. Just a few of them are hair protein. So, your hair is dead cells, but it is made out of different types of proteins, as are your muscles, and as is your skin and your eyes. Now, protein synthesis is going back to this thing you're going to see in your future biology classes. It's called the central dogma. So basically what it says is your DNA gets turned into something called RNA, which carries the message of DNA to your protein. So DNA to RNA to protein. And the two steps of protein synthesis are transcription and translation. And something to remember this, or help you remember this, is that if you've ever heard of the book called The Odyssey or the Iliad, they were written by an orator named Homer. And orators in the Greek tradition would basically just tell these long epic stories. And so basically Homer had this thing memorized and he would just say the story. And people liked it so much that they would write it down. So you got a guy speaking the story, you would write down or transcribe the story so people could remember it after Homer kicked the bucket. And then, what if you don't speak ancient Greek? What if you speak a different language? Well, then we've got to translate it so people all over the world can read the transcript. So that's basically what is happening in your cells. What happens is that the DNA is the message, right? It's kind of like Homer telling the story. Well, the DNA needs somebody to write it down. So this thing called RNA writes down or transcribes DNA's story, and then it's translated into a protein so that your body can basically understand it and build. Now, what is this RNA stuff? So RNA is a nucleic acid that's very similar to DNA. In fact, if you want a good visual, here's what they look like. There's a few key differences. RNA's only got one side of a double helix, so it's single, and it has a cytosine, a guanine, an adenine, and a uracil. The only difference is that it has a uracil instead of a thymine. So they're both the same type of base, but they're a little bit different. As you can see, there's a methyl group, a CH3, right there, that uracil does not have. Okay, so that's how RNA is different. It has one half of a double helix, and it has a uracil instead of a T. It works the same way as DNA does. For example, if we pair up a guanine with RNA, it's going to go to a cytosine just like DNA would. And if we're going to pair a guanine of RNA, it's going to go to a cytosine of DNA. The only difference is that instead of A to T in DNA, it's going to be A to U. So if we have a T in DNA, it is going to still pair up with the A like it normally would. But if we have an A in DNA, it would normally pair up with a T. But we don't have a T in RNA. It can't say T. It can only say U. So anytime you have an A in DNA, it goes to a U in RNA. That's the only difference. So if we write that out, we would say G to C, T to A, C to G, and A to U. And the last difference between RNA and DNA is that it has a ribose instead of a deoxyribose. So the oxygen is on that ribose sugar, that pentose sugar in the nucleotides. So that's why it's called RNA, because it's ribonucleic acid, not deoxyribonucleic acid. The most important differences are the U and that there's only one strand. So, moving on, we have a couple examples of America's Greatest Wingman. And back to the central dogma. So, we got our DNA. DNA is the storyteller. RNA is the scribe that writes it down. And then there's another type of RNA that is actually going to translate it, and that is going to be translated into protein. And here's how it actually happens. So if we get rid of that little mess, we have our DNA, and it's being transcribed into RNA. So this is where the story is being written down. This all happens in the cell's nucleus. So if this is a cell, that happens in the nucleus. And then the mRNA is also a messenger. That's what the M stands for. It's messenger RNA. So this is not only a scribe, but it's a messenger. It actually takes the message outside of the nucleus into the cytoplasm. Remember the outer region outside of the nucleus? And it's going to take it to a protein. Excuse me, it's going to take it to a ribosome, and it's going to make it into proteins that make you. So that's what it's actually saying. Now, that was in a prokaryotic, so I drew the nucleus in there for what would be in a eukaryotic cell, and there's your picture. So that's basically what we just did. Now, our pathway to making a protein is DNA to RNA to protein, and this is the messenger and the scribe, and then this is the guy that's actually going to translate it. Okay, the T does not stand for translator, it stands for transfer, but that's what it does. So let's go ahead and write ourselves a note. Messenger and the translator. And it happens in the protein. So step one, transcription. So here we have DNA. And this is happening in DNA replication, which we didn't cover this year. But here's the important thing. So DNA, we have a C, A, T, G, G, C, okay? So let's practice our transcription into messenger RNA. So C is going to go with a G. A can't go with T because mRNA does not have a T, so this is going to go to a U. Again, A goes to a U, T goes to A, G goes to C, and G goes to C, and C goes to G. So that is our example transcription of mRNA. Then it's going to take it out of the nucleus. And it's going to look for a ribosome. So here's our nucleus. mRNA takes DNA's message outside, and it finds a ribosome protein. This is where translation is going to happen. So transcription is in the nucleus. Translation is outside of the nucleus. And mRNA is going to be read 3 bases at a time. So let's go back up and see what actually that would look like. So if we look at this mRNA, let's compartmentalize it. It's read 3 bases at a time. So when it's going to be translated, it's going to be read into that segment, and then in that segment, and so on. So it's going to be broken up into 3s. These 3s, triplets, are called codons. So like if we have AUG, GUC, CCA, what we have are 3 codons. That's a codon, that's a codon, that's a codon. So all it is a triplet of RNA nucleotides. So now, how is this translated? So here's our messenger RNA that's come out of the nucleus into the cytoplasm, and this was the message that it brought. So it brought us a GUC, that's a codon, that's a codon, and that's a codon, that's a codon. You get the picture. Well, how is it translated? This is what tRNA looks like. It's this big hunk of thing. And it has two important pieces. The point of attachment, which is called the anticodon right there. All it is the complementary RNA to the mRNA. So what goes with a G? C. What goes with an A? U. What goes with a C? G. And so it uses the mRNA to pair up at the site of the anticodon. So it's kind of like a stamp. Think of it that way. Okay, so this is the envelope or the paper, and this is where it stamps on. Now, the anticodon is important because there's different types of tRNA that all have different anticodons. So that way we have like different stamps that match up to different pieces of paper. Well, each stamp where you would press down carries something called an amino acid. These are the building blocks of proteins. So if a protein is like a string of beads, each bead is an amino acid. So each one of these guys, if that is the protein, that's an amino acid. So here is another picture of it. Here we have our ribosome. So let's recap. The mRNA took DNA's message in transcription. Okay, that happens in the nucleus of a cell for a eukaryotic organism. DNA goes to RNA. Then it leaves, so the mRNA takes it out, finds a ribosome, which is here we are, and this is where translation happens. So here's our ribosome, here's our mRNA, and here's our first codon, this triplet AUG. This paired up with this tRNA that had an anticodon of UAC. And UAC was able to bring this MET start amino acid. So this is our first amino acid. There's actually a total of 20 of them. I'll show you how that works. But then here's our next one. So now our next codon was UUA. And AAU is our anticodon, so that pairs up. And that means that that tRNA is attached to a LU, or leucine amino acid. And then we have glycine right here, which is going to be coming up next. What do these tRNAs do? After they stamp, they shift over here, and then they leave to go get more amino acids and take them back. Then this guy is going to bond next and put on the glycine. And then it keeps going down the entire stretch of mRNA. So here's what we're going to do. If we have a sequence of mRNA, how do we read the protein sequence? It's quite simple. We have an amino acid key right here. So what we're going to do is we're going to look at our first one. Our first codon is AUG. Your first base is the A for AUG. So we go to the A, and that means that our amino acid protein is going to be somewhere in this column. So we're going to highlight, this row. This whole row. It could be any one of these. Well, then we get more specific. So we know our next base is a U. And so then we have now localized it to this column and this row. Then what's our third base? The G, AUG. Well, now we know that it is not only in that box, that series of 4 boxes, it is actually that box. So you just plug in the first base, the second base, first, second, and third, and you just grid line it to where they all intersect. So let's do the next one, CGC. So CGC, here we go. C, it's going to be anywhere in this row. G, it's going to be somewhere in there. And C is our third base. It is arginine. So we have met, arginine. And then we have THR, which is tyrosine. So that's how we do this. Now, when we go back to our mutation slides real quick, mutations, let's say we had like a frame shift mutation. So what was our original DNA? Well, what would go with an A? Let's say we had T. So our DNA, our original DNA, let's work backwards. A goes to T, U went to A, G went to C. Then it was C went to G, G went to C, G went to C, and A went to T, and C went to G, and C went to G. Now what happens if we have a frame shift? So let's say when the DNA got copied, we instead got an extra G right there. Well, here's what that would cause. Our first codon would be the same. Then that G would push everything over. So then it would be G, G, C. And then that C would be pushed over to C, T, G. So now let's go through and see what our amino acid sequence would be. If we change this, it's still AUG for mRNA. Then what happens? G would go to C, G would go to C, C would go to G, and then C would go to G, T would go to A, and G would go to C. See how now we have two different codons because of that one G getting pushed, pushing everything over that way. So now we have different proteins, which means we have a different use. So that would be MET. What's CCG? So C, C, G, proline. What's GAC? G, A, and C, aspartate. So notice that with one mutation, we have changed all the proteins down the road. So we've changed the entire sequence continuing after the MET amino acid. Different amino acids means you have a different protein. It's like having different colored beads on a string. In this example, we could have a red bead, and then an orange bead, and then a yellow bead. But with the mutation, we started out with a red bead, and then what happened after that? We got green and blue. So this is how mutations actually cause differences in your proteins because of this. And so this is just kind of a review of how mRNA works. And it's showing you the difference in amino acids. And then this is a review of central dogma. So DNA is where the message tells the story. It is transcribed into mRNA. This is our scribe who writes down the message. Then it takes it out of the nucleus to a ribosome, and our tRNA is the guy who translates it into proteins. So DNA to RNA to protein. So what you're going to need to know, and this we're going to practice this, is how DNA is transcribed into mRNA. mRNA takes it out of the nucleus to a ribosome, which is where it is translated by tRNA into proteins. So remember, the triplets of RNA are codons, and amino acids are building blocks of proteins, or like beads on a string.